My background was in genetics, genomics, molecular biology, and I was tasked with finding the mutation that causes the scurfy phenotype. And we decided to take a two-pronged approach to cloning scurfy that would involve first genetic mapping. So we knew the scurfy locus was on the X chromosome and some sort of general localization had been performed already. So we had a good starting point. But I decided that we would just continue to let the mouse do the work for us and start trying to narrow that interval further by scoring for recombination events that would help to narrow the scurfy locus. So we brought the scurfy mice to Darwin in Seattle, but first we had to find a home for them, and so that was an underutilized janitor's closet with a sink and a self-contained mouse caging system. And I was able to set up a big backcross experiment in that janitor's closet in order to score recombination events. And this is just a copy of a piece of paper that I had up on my bulletin board for years, it seemed like, where I was tracking recombination events and also trying to compare our X chromosome mapping to other maps that were being published at the time. It was a very confusing time where there wasn't, you have to understand that there was not really genomic sequence available at the time, and so but everybody was really trying to put together maps showing orders of markers, etc. So we combined the genetic mapping with physical mapping. So we wanted to ultimately really understand at the DNA level, at the nucleotide level, what was happening in the region that contained the scurfy locus. And as I said, as sort of an outcome of the Human Genome Project, there were a number of really, cutting-edge technologies that were available, and some of these are listed here. And I think it's worth pointing out that our situation was perhaps unique. Being in a biotech company, we had the funding that allowed us to incorporate a lot of these different resources, as well as the mouse genetic breeding experiment as well, which is all very expensive. It would be difficult for an academic lab to do all of this simultaneously. So the process that we went through for finding the scurfy mutation involved, as I said, the genetic mapping. We ultimately found, so the red X's on this chromosomal region represent the most informative recombination breakpoints that we identified. And at the same time that we were doing the genetic mapping, we were using whatever molecular markers we had in that region to start pulling out large cloned fragments of genomic DNA and kind of generating a tiling path or a contig of artificial chromosomes across the region. And in the end, we were able to estimate that the physical region was about 500,000 base pairs long. And once we had a number of cloned sequences across the region, then we started doing really high quality DNA sequencing of them. So we had a really good core facility to do the DNA sequencing ourselves in-house. Then once we had that really high quality genome sequence across the region, we were able to apply whatever kind of analytical tools and algorithms that were available at the time. And we ended up with a list of about 20 genes in the regions. And then the trick was just to sequence, and this was all sequence, of course, that was derived from the wild type version of genomic DNA. And so for each of those genes, we cloned and sequenced the scurfy version of the gene, looking for a mutation. And where we found the mutation turns out to be this gene here that we were calling FKH. It was a novel gene that had never been reported, but it did have homology to a family of fork-head domain containing DNA binding proteins. And this is a page out of Catherine's lab notebook that shows that reports the mutation. This slide here is kind of our overall summary then of the region, which shows the gene content. Again, these X's represent the most informative recombination breakpoints. And then I wanted to show this nice sequencing chromatogram, because we never look at those anymore, that clearly shows an insertion of 2 extra adenines in the scurfy versus wild type. And this, this mutation, of course, would cause a frame shift in the translation of the mRNA. And it's located in the DNA binding domain of this novel fork head protein.